Have you ever stopped to think about what's really going on behind the scenes in your brain? What's driving your motivation to get things done? Or what's coordinating the simple act of picking up your coffee? Or even what's setting the entire mood for your day? Well, the answer isn't some abstract concept. It's chemical. It all comes down to a tiny but incredibly powerful group of molecules called monoamines. So today, we're diving deep. This is a masterclass on these critical chemical messengers, dopamine, noradrenaline, and serotonin, so we can really understand the intricate machinery that shapes our reality. Okay, let's get right into it.
So, here's our game plan. First, we're going to get the lay of the land. Map out the major neural highways these molecules travel on. Then, we'll follow the entire life story of dopamine, our so-called molecule of motivation. After that, we'll see how everything we learned about dopamine gives us a perfect head start for understanding noradrenaline. Next up, we'll jump into the unique world of serotonin, the mood setter. And to wrap it all up, we'll connect all the dots, looking at some really powerful genetic evidence to see how this chemistry translates directly into behavior.
All right, so before we can really dig into the molecules themselves, we need a map. We have to understand the specific circuits in the brain, kind of like superhighways where they actually do all their work. Let's start by getting our bearings and taking a look at the major pathways of the dopaminergic system. Now, what's so cool about this is how it neatly organizes dopamine's huge influence into three main systems. First, let's trace the nigrostriatal pathway. Think of this as the brain's master circuit for movement. When this pathway degenerates, it is the direct cause of the tremors and stiffness you see in Parkinson's disease. Second, you've got the big one, the mesocortical limbic pathway. This is where motivation, reward, and our executive functions live. When this system gets out of whack, it's tied to just about everything, from schizophrenia and addiction to ADHD. And third, we have the tuberoinfundibular pathway. This is a more specialized circuit that regulates hormones, and when it's disrupted, it can lead to conditions like hypoprolactemia.
Okay, so with our map in hand, let's start our deep dive into the world of dopamine. We are going to become its biographer. We're going to follow its entire journey from the raw materials it's made from to how it's packaged and stored, how it's released, and what it does, and finally, how it's retired and broken down.
To really get a sense of how important dopamine is, just look at this number, 80%. Think about your brain's dopamine supply like it's a full reservoir. Now, over a lifetime, all of us naturally lose about 10%. It's a slow decline. But Parkinson's disease? That only shows up when there is a catastrophic leak, a just devastating loss of 80% of the dopamine-producing neurons in that critical region, the substantia nigra. It's a terrifying tipping point. And it's not just about loss. An imbalance or dysregulation of dopamine is also a key factor in the complex symptoms of schizophrenia and ADHD.
So, how does the brain actually build this crucial molecule? Well, as you can see in this pathway, it all starts with a simple amino acid, tyrosine. It gets pulled into the neuron with a little help from a cotransporter that also brings in a sodium ion. Now, the most important step, the main bottleneck in the whole assembly line, is controlled by an enzyme called tyrosine hydroxylase, or TH. This enzyme turns tyrosine into a molecule called L-DOPA. But to do its job, TH needs two little helpers, a cofactor called tetrahydrobiopterin, or THB4, and vitamin C. And the fact that there's such a high concentration of TH in the nigrostriatal tract is actually how we identify these dopamine neurons. Finally, a second enzyme, dopa decarboxylase, steps in. It snips a carboxyl group off of L-DOPA to create our final product, dopamine. And this step needs its own helper, pyridoxal phosphate.
Let's just zoom in on that gatekeeper enzyme, tyrosine hydroxylase, for a second. Its activity isn't set in stone. The neuron can actually ramp up dopamine production by slapping a phosphate group onto TH, and that process depends on calcium levels. This just shows you how crucial this one enzyme is. There's a fascinating and pretty tragic clinical story from some Amish communities that really brings this home. Because of high rates of inbreeding, a specific mutation in the gene for tyrosine hydroxylase has popped up. This faulty version of the enzyme just can't produce dopamine as well, and the result? A chronic dopamine deficit that has been directly linked to a much, much higher risk for depression in that population.
Once dopamine is made, it can't just hang around inside the cell. It's a really potent chemical, and it needs to be stored safely. This is the job of synaptic vesicles, and this amazing little molecular machine called the vesicular monoamine transporter, or VMAT. The way it works is just brilliant. Think of the vesicle like a rechargeable battery. An active pump uses energy, from ATP, to cram protons into the vesicle. This creates a really powerful proton gradient. Then VMAT, our cargo loader, taps into that energy potential. It swaps one proton out for one dopamine molecule in. This whole process can be blocked by the drug reserpine, which pretty much just drains the neuron of all its stored dopamine.
Okay, now for the action.
When dopamine gets released, its message can either be a go signal or a stop signal, and it all depends on which type of receptor it docks with. They basically fall into two opposing families. On one side, you have the D1-like receptors, that's D1 and D5. Think of these as the go switches. They're hooked up to a stimulatory G protein, GS, which then activates an enzyme and revs up production of a key messenger called CAMP. On the other side, you've got the D2-like receptors, D2, D3, and D4. These are the stop switches. They're coupled to an inhibitory G protein, GI, which does the exact opposite. It shuts down that enzyme and lowers CAMP levels.
I know this table looks a little dense, but it tells a really crucial story about diversity, and maybe more importantly, location. We don't need to memorize every single detail, let's just focus on the big picture. The key takeaway is in the distribution column. The D1 and D2 receptors, which are the most common, are packed into the motor control centers like the caudate and putamen. That's why dopamine is so critical for movement. But then look at the D4 receptors. They're found in the frontal cortex, the headquarters for our executive function. And D5 receptors are in the hippocampus and hypothalamus, which are crucial for memory and our basic drives. You see? This very geography is exactly how one single molecule can play so many different roles all over the brain.
After dopamine has delivered its message, it needs to be cleared out and broken down. This diagram shows us two parallel pathways for its degradation, and both of them involve two key enzymes, monoamine oxidase, or MAO, and catechol-O-methyltransferase, or COMT. Just think of it like two different roads that both lead to the exact same destination. In path number 1, MAO acts first, then COMT follows. In the other path, the order is just flipped. But notice, both routes in metabolism end up at the exact same final product, homovanillic acid, or HVA. And this is the critical point. Because HVA is the final, stable end product, clinicians can measure its levels in your cerebrospinal fluid to get a really reliable snapshot of the dopamine activity happening in your brain.
So how does the neuron actually turn the signal off in the synapse? Well, the main method, which handles about 75% of the cleanup, is called uptake 1. This is the job of a molecular vacuum cleaner called the dopamine transporter, or DAT, which literally sucks dopamine back into the same neuron it just came from. And this is the exact transporter that is famously blocked by cocaine, which causes dopamine to hang around in the synapse and creates its powerful effects. The other 25% is handled by uptake 2, where nearby glial cells called osteocytes absorb the dopamine to break it down. Now, once dopamine is back inside the neuron, it has two possible fates. It can be recycled and repackaged into vesicles by VMAT to be used again, or it can be destroyed by the MAO inside the cell. And that destruction process actually produces hydrogen peroxide as a byproduct, which can contribute to oxidative stress and cell damage over time.
Okay, now for a true story of neuroscience detective work. A real tragedy that unlocked a profound secret about Parkinson's. It all starts with a chemical called MPTP, which was a contaminant in a bad batch of synthetic heroin. MPTP itself is like a silent assassin. It's harmless enough that it can slip right past the blood-brain barrier. But once it's inside the brain, astrocytes convert it into a truly toxic molecule, MPP+. And here's the diabolical twist. The dopamine transporter, DOT, mistakes MPP+ for dopamine and actively pumps this poison into the very neurons it's supposed to be protecting. Once it's inside, MPP+ launches a direct assault on the mitochondria, shutting down energy production by blocking something called complex I. This starves the neurons of ATP, leading to rapid cell death and causing permanent, irreversible Parkinson's symptoms in the young people who were exposed to it.
So how can we use medicine to manipulate this system? Well, if we need to turn down dopamine signaling, like for treating psychosis, we can use D2 antagonists like haloperidol. They just block the receptors and turn down the volume. On the flip side, D2 agonists like bromocriptine actually stimulate the presynaptic autoreceptors, which tricks the neuron into releasing less dopamine. To protect the system in Parkinson's, we can use MAOB inhibitors like racegiline, which prevent dopamine from being broken down, making more of it available. And of course, the gold standard treatment for Parkinson's is L-DOPA. This is such a clever therapy because it bypasses that rate-limiting TH enzyme completely by just giving the brain the next molecule in the chain. It's always given with a drug called carb-DOPA, which stops the L-DOPA from being converted into dopamine in the body before it has a chance to reach the brain.
All right, now that we have a really solid grip on dopamine, let's move on to our next monoamine, noradrenaline. And here's the really cool part. We don't have to start from scratch. As we're about to see, the story of noradrenaline is basically the direct sequel to the story of dopamine. Its synthesis begins right where dopamine's ended. So the life cycle of noradrenaline, or NA, has some really key differences. The synthesis step is fascinating. It actually happens inside the synaptic vesicle. Dopamine is first packaged into the vesicle, and only then does an enzyme called dopamine beta hydroxylase, which also needs vitamin C, convert it into noradrenaline. It's stored in these special vesicles, which are physically denser than the ones that carry other neurotransmitters like glutamate. Its release is dependent on calcium, but it happens from these widespread nerve endings called varicosities, and it has a pretty low probability of happening. And finally, the cleanup is handled by its own dedicated vacuum, the norepinephrine transporter, or NET, which recycles about 75% of it.
Noradrenaline acts on a whole family of adrenergic receptors. As you can see in this table, alpha-1 receptors, which are targeted by drugs like phenylephrine, cause vasoconstriction. They tighten up blood vessels. Alpha-2 receptors often act as autoreceptors, which means they provide negative feedback to the neuron by lowering CAMT levels. The beta receptors, on the other hand, usually increase CAMP. So beta-1 stimulation revs up your heart rate and makes it contract harder, while beta-2 stimulation, which is targeted by the common asthma drug salbutamol, causes vasodilation. And what's really interesting here is a huge Norwegian study that found a surprising link. People who regularly used the beta-2 agonist salbutamol had a significantly lower risk of developing Parkinson's disease. It's a hint that this pathway might have a protective role.
And now for our final protagonist, the famous mood molecule, serotonin, also known as 5-HT. So for this last part, we're going to explore its unique origin story. Unlike the others, which are made from tyrosine, serotonin is built from the amino acid tryptophan. It's produced mainly in the brainstem's Raphe nuclei, and we'll see how this one molecule influences everything from our appetite to our sleep.
The synthesis pathway for serotonin should look pretty familiar by now. We start with tryptophan. The rate-limiting enzyme, which is called tryptophan hydroxylase, adds a hydroxyl group, and it also needs TBH4 to work. Then, a decarboxylase removes a carboxyl group, and boom, you have serotonin. But look at the second half of this diagram. It reveals another critical role. In the pineal gland, serotonin isn't the end of the line. It actually serves as the direct precursor for melatonin, the master hormone that governs our circadian rhythms and tells our body when it's time to go to sleep.
Just like dopamine, serotonin's effect is all about which receptor it binds to, and its receptor family is incredibly diverse, with 7 main branches. As this diagram shows, some of these receptors are inhibitory. They couple to GIO proteins to quiet down neural activity. Others are excitatory. They couple to GS or GQ proteins to ramp things up. This massive receptor diversity is what allows a single molecule to regulate such a huge range of functions, from mood and anxiety to things like digestion and even nausea.
This slide is a fantastic illustration of the battlefield where so many powerful drugs do their work. The most famous, SSRI antidepressants, work by throwing a roadblock right here at the SERT transporter, which stops serotonin from being taken back up. MAOIs prevent it from being broken down inside the neuron. The hallucinogen LSD produces its profound effects by targeting the 5-HT2A and 2C receptors. And the potent anti-nausea drug ondansetron works by blocking 5-HT3 receptors. It's also really important to point out the neurotoxic effects of long-term ecstasy or MDMA use. It can cause severe, lasting damage to these very serotonergic neurons, which can potentially lead to chronic depression and cognitive issues down the road.
Okay, so we've learned a ton about the intricate molecular machinery of these monoamines. But how can we be absolutely sure that these specific molecules are responsible for all the vital functions we've been talking about? In this final section, we're going to look at some fascinating experiments that give us the ultimate proof by seeing what happens when you just remove one of these critical parts from the system entirely.
And this table just brilliantly lays out the consequences. This isn't just a correlation, folks. This is a direct demonstration of cause and effect. If you knock out the gene for tyrosine hydroxylase, the very first enzyme in the dopamine pathway, the mice are simply not viable. They can't survive. If you knock out the D2 receptor, the mice show Parkinsonian-like motor problems. If you take out the dopamine transporter, DAT, you get hyperlocomotion, a state of constant restless movement. If you remove MAOA, the mice exhibit intensely aggressive behaviors. These knockout experiments give us undeniable organism-level proof of the specific and essential role that each and every one of these proteins plays.
So we end our journey here, having gone all the way from single amino acids to the vast complexity of behavior. And it leaves us with a profound final question to think about. How does this intricate, beautiful chemical machinery actually give rise to our thoughts, our feelings, and our actions? Where does the chemistry end, and where does the self begin?